Friends, welcome to Biology Made Easy. Today we'll discuss mutation. So you know that mutation is a sudden change in the genotype of the organism, and it leads to either qualitative or quantitative changes which is also reflected in the phenotype. So one thing you should remember, since it changes the genetic material, the genotype, it must be heritable. And another important thing, mutation is the source of all types of variations. When we don't get a variation, then we can go for mutation. And by mutation, we can get variations, and these variations are the raw materials for evolution. So today we'll discuss about this mutation. The term mutation was first coined by Hugo de Vries. When you worked on Oenothora lamarckiana. Now, what are the different types of mutations? Basing upon the cell type, we can tell that it may be germinal or it may be somatic. When we'll call it a germinal? When the mutation takes place in the reproductive cells, in the germ cells during production of eggs and ova. So that is known as germinal. That means when there is germinal mutation, it must be it is heritable from the parents to the offsprings. But when it takes place in somatic cells, in the body cells that do not take part in the reproduction, that means it is not heritable. So what is the example of the somatic variations? There are very good examples. One is Naval orange. This is the name of one variety. And another is delicious apple. So these two, orange and apples, they have been produced by means of a somatic mutation. Now one question may come to our mind, since they are not heritable, then how can we grow generation after generation? Then the answer is by vegetative means. We can propagate them by vegetative means. So depending upon cell type, these are the two types. Now, depending upon the origin, the mutation can be of also two types. One is spontaneous and the other is induced. Now, what is the meaning of spontaneous mutation? Sometimes in the nature, the mutation takes place automatically. The reasons are environmental reasons. And when artificially we induce mutation, then it is known as induced mutation. What is the necessity of this induced mutation? Suppose we want a variation and that variation is not found in nature, then we can try for mutation, we can go for mutation, because by mutation we can get the variations. So how will induce mutation? Well, either physically or chemically applying different substances. So the substances which are applied to induce mutation, they are known as mutagens. So this is again important for examination point of view. What are mutagens? That means substances required to induce mutation are known as mutagens. Now let us have a discussion about these mutagens. The mutagens, they are classified mainly into two categories. They may be physical mutagens or they may be chemicals. The physical mutagens, these are again of different types. They may be ionizing radiations, and what are the examples? Example, it may be alpha ray, beta ray, gamma ray, X ray, all these are. And one scientist known as Muller, he is the first person to use X-ray in Drosophila to induce mutations, the first scientist. And when we apply ionizing radiations like X-ray, gamma ray, all these things, what happens? In the living tissues, when we give X-ray or gamma ray, the living tissue, the different atoms they are present inside the living tissue, they got ionized. And ultimately, there is a break in the DNA, that leads to mutation. There may be deletion, there may be addition, there is a breakage, all these things will take place. Then come to the second one. The second one is non-ionizing radiation. And among the non-ionizing radiations, one important thing is UV light. The other is microwaves and also radio frequency. Out of these things, commonly this UV light is used for inducing mutation. How it induces mutation? Suppose these are two strands of the DNA, and here thymine is there, here also thymine is there, then this side it will be adenine. When we apply UV radiation, usually two adjacent thymines they form dimers, thymine dimers. As a result, what happens? The entire DNA bends down, and when these two they give rise to dimers, they cannot combine with the T, so it will lead to changes. It leads to mutation. Then the third one is temperature. So in case of temperature, what happens? When there is an increase in temperature, usually it leads to denaturation of DNA. But in one case, when there is a decrease in temperature, particularly in case of rice, when there is a decrease in temperature, usually the number of mutations increase. Mutations increase in a low temperature in case of rice. But in other cases, it leads to the denaturation of the DNA molecules. So these are the different physical mutagens. Then let us discuss the chemical mutagens. So the chemical mutagens are also of different types. They may be nitrous acid, may be alkylating agents, may be base analogues, or they may be acridins, they may be mustard gas, or they may be even colchicine. Now, number one is nitrous acid. This is one of the very important mutagens. Usually the nitrous acid acts as an alkylating agent. It acts as deamination. It acts as a deaminating agent. So when it helps in deamination, what happens? Now the cytosine is converted to uracil. The guanine is converted to xanthine, and adenine is converted to hypoxanthine as a result of deamination. So when these changes take place, what happens? Suppose in one case this C pairs with the G. Now this C will be converted to uracil. So now uracil will not combine with the G, it will combine with the T. And in the next replication, the T will combine with A. So originally it was CG, now it is converted to AT. In this way mutation takes place. In all these cases, it takes place like this. This is number one. Now the number two is alkylating agents. Alkylating agents. So alkylating agents usually they cause ethylation and methylation, either ethylation or methylation. Either they cause ethylation or methylation of the different substances. So due to this ethylation or methylation, the pairing behavior of the nitrogen changes. And what are the common alkalic agents? They may be ethyl ethane sulfonate, ethyl methane sulfonate, methyl ethane sulfonate, or methyl methane sulfonate. So these are the different alkylating agents. They cause mutation. Then the third one is base analogues. So what are the base analogues? Base analogues means there are some substances inside this cell which are similar in their structure to that of the nitrogen bases. And one such example is 5-bromouracil. 5-bromouracil, which is structurally similar to that of thiamine. So suppose in one case this thiamine combines with A. So now this thiamine will be changed to 5-bromouracil. Then it will not combine with A. It will combine with G. And in the next replication, this G will pair with C. So you see originally it was TA, but finally converted to CG. So in this way the changes take place. The fourth type is acridins. Acridins are usually aromatic compounds which are used for preparation of dyes and pharmaceuticals. When such substances are used, usually they cause deletion or insertion of different compounds leading to mutation. Then the fifth one is mustard gas. Mustard gas, it may be nitrogen mustard or sulfur mustard. It also causes break in the DNA. And the another is colchicine. Colchicine usually what it does to the organism, usually it doesn't allow the spindle fiber formation. During cell division, the spindle fibers which are formed, they are not allowed to form. As a result, the number of chromosomes, the chromosets are multiplied. So these are some of the different mutagens, both chemical and physical mutagens. In addition to this, there are also certain biological mutagens, like some viruses. Some viruses also, they cause changes in the behavior of the genes. And another biological agent is transposon. Transposon or jumping gene discovered by Barbara McClintock in case of corn. So since these are jumping genes, they jump from one chromosome to another chromosome, so they can also cause mutation, but these are biological mutations. So in this way, these are the different mutagens that we have discussed.